and I think it's sort of a long ongoing discussion, and it's something that was talked about previously, which is the performance of inactivation buffers for PCR reagents. So, Britta, up to you.
Right. Thanks, Don. So as you mentioned, this is driven by some previous work from other labs, and this is still an ongoing project, so we'll see what happens with the rest of my results. So to start off, I think everyone in this room is aware that there are very tight biosafety concerns and regulations surrounding the handling of foot and mouth disease, and it's critical that the lysis buffers used in these labs are effectively inactivating the virus. And this is important for determining the location of where nucleic acid extractions are being done, as well as for the external transfer and shipment of samples. So for example, can you take your samples in lysis buffer out of high containment to run your PCRs? So the purpose of our project was to determine whether a selection of commercially available lysis buffers are able to inactivate FMDV effectively, and this is indicated by the lack of CPE or virus replication in cell culture. So just to go over my experimental design briefly, more details are on my poster, but we selected 3 lysis buffers, and these are ones that we are currently using in the lab or ones that we are hoping to switch to. So the first is a lysis buffer from the MagMax kit, the second is a MagVet kit, and then the RNA Easy kit. So far to date, we've tested a virus from all 7 serotypes. The majority of these have been cell culture supernatants, so these are having high titers of virus, and we also use one that's an original suspension. The titers that we are putting in with the lysis buffer are ranging from 6.9 to 8.5, so we wanted to challenge the system with the highest titers possible. And I should say that the viruses that we're using are recent strains coming from the field. For our experiments, we're using 2 different cell lines, primary bovine thyroid cells, which are highly sensitive to FMD, along with a continuous pig cell line, or IBRS2 cells. In our experiments, we're inoculating cells and incubating them for 3 days and observing for CPE. If we don't see any CPE on this first passage, we're doing a blind passage, so we're freeze-flying our samples and then inoculating again for another additional 3 days. And so far to date, what we've found is that the lysis buffers that we're using in the lab are inactivating FMDV, as indicated by the lack of CPE in cell culture. And so far, this is only for cell culture supernatant and original suspensions. We do have a number of future experiments that we will be conducting. One is to do similar experiments for SVD, which is clinically similar to foot and mouth disease. And we also want to investigate different sample types, such as probang, milk, plasma serum, and then feces for SVD. So with that, thank you for your attention.
Thank you, Britta. It's very clear. Any questions? So I can immediately see there is a question. Maybe while you're bringing the mic, Emmy has one question as well.
This is just quick. So how do we relate to the issue of full-length RNA? So infectivity is gone. How will the full-length RNA, will there still be full-length RNA in the samples? And do we consider that a problem? That's more to the reference laboratory.
I'm not sure I fully understand the question. I'm sorry.
Are you asking whether the lysis buffers that are used have an impact upon the quality of the RNA that's subsequently?
No, that's not what I'm asking. I'm asking, so we're looking at this because we want to ascertain that the viruses are not viable after having been in lysis buffer, because we want to export them to less contained areas. In our laboratory, we have an issue with full-length RNA that we don't want to take out of the FMD laboratory either. So the question is, these samples that have been in lysis buffer, will there be full-length RNA in them? And does the World Reference Laboratory now recommend that that's not a problem?
So we haven't been testing for full-length RNA out of these samples, so I can't directly answer that question. If Don has any additional
we'd
input.
anticipate there would be full-length RNA in the samples, and that's the whole purpose of the lysis buffer. So it's not a direct concern for us because we're doing all of this work at BSL-3+. So we're not doing this work to show that subsequently you can handle that RNA at lower containment. For me, that's a slightly different discussion.
I just wanted to have that clarified.
Yeah.
ask another question? You have used the lysis buffer on tissue suspensions. Have you also checked with epithelium tissues?
Yes, so we've tested one that's original suspension. We will be doing further experiments, but this is how far we've gotten to date.
Okay, thanks, Britta. So do you want me to...